Hi friends, welcome to my video. In this video, we are going to see about antibodies. What are antibodies? Antibodies are epitope binding glycoproteins produced by plasma cells in response to antigens. Let's see, what is an epitope? Epitope is the part of an antigen that is recognized by the immune system. For example, epitope is the part of an antigen molecule to which an antibody attaches itself. Next, we'll see what is glycoproteins. Proteins with attached carbohydrates are called glycoproteins. Next, we'll see what are plasma cells. Plasma cells are white blood cells that develop from B lymphocytes or B cells, a type of white blood cells. B cells, after encounter with antigen, differentiate into specific antibody secreting plasma cells. Let's see the definition once again. Antibodies are epitope binding. That means antibodies bind to the epitope of antigens. Glycoproteins produced by plasma cells in response to antigens. We know antigens are any substance which, when introduced into the body, stimulates the production of an antibody. Antigens may originate from within the body or from the external environment. For example, bacteria, viruses, pollen, some chemicals, fungi, etc. An antibody is also known as an immunoglobulin. Let's see, what are immunoglobulins? Antibodies are termed immunoglobulins because they are globular proteins with immune function and most of which generally separate in the gamma region on electrophoresis. Immunoglobulins are Y-shaped glycoproteins, each molecule consisting of two pairs of polypeptide chains of different sizes. The smaller chains are called light chains and the larger chains are called heavy chains. The antigen combining site of the molecule is at its amino terminus, which is composed of both light and heavy chains. There are 5 separate classes of immunoglobulins. They are IgG, IgM, IgA, IgD, and IgE. The most common antibody in serum is IgG. The largest immunoglobulin is the IgM. The predominant immunoglobulin in external secretions such as saliva, tears, etc. is IgA. IgE are responsible for hypersensitivity or allergic reactions. Antibodies are found in two forms, as either membrane-bound constituents of B cells or as soluble molecules secreted by plasma cells. Membrane-bound antibodies confer antigenic specificity on B cells. That is, when a specific antigen is present in the body, membrane-bound antibody allows a B cell to detect it and triggers B cell activation. Secreted antibodies circulates in the blood where they attack and neutralize antigens that are identical to the one that triggered the immune response. Next, we'll see about some important functions of antibodies. Antibodies prevent infection and disease caused by the microorganisms. Antibodies react against specific microorganisms and their toxic products. Antibodies can be used in the treatment of infection caused by the specific microorganisms. Next, we'll see what is immune sera. Sera having high antibody levels following infection or immunization were called immune sera. I am concluding this video here and see you soon in my next video. Thank you, friends.